I'm curious about how much useful chemistry already exists in biology that we haven't learned how to access yet. Hi, I am Karsten Eastman. I am the co-founder and CEO of Sethera Therapeutics. We are a biotech based in Salt Lake City, Utah.
Can you tell me more about what Sethera is doing and what are the therapies?
Really, Sethera that sits at this intersection of chemistry, biology, and drug discovery. That's the really big picture. And at the simplest level, what we are doing is we are layering enzymes on top of everything that we do. So enzymes allow us to build molecules that are difficult to truly impossible to make through traditional synthetic chemistry. An enzyme is a protein. A protein is made of amino acids, and peptides are also made of amino acids. So that's where you can get some of the really complex interactions that peptides can have in biology. They're just a small protein, but that means that there's all of this lovely biological space that they can exist in. We're first focused on building a platform that uses enzymes to hyper transform peptides into more complex, highly structured molecules called macrocycles and polymacrocycles. These can act more like an antibody in terms of, say, binding very large, complex spaces, but they can be much smaller than an antibody and have more drug-like properties of small molecules. The broad goal that we have is to use this enzyme platform to create new types of therapeutics that can reach targets or indications that have been historically very difficult to drug.
So you're saying you use enzymes to make peptides.
That's a good way to think about it, but we use several different enzymes in the process. So one of the enzymes uses the natural machinery to produce the peptides from genetic information, but then we use another enzyme on top of all of that to take a linear peptide, which is floppy, it can move around, and then conformationally constrain it, so basically lock it into place, so it will hit the desired area and stay on the desired area. Peptides themselves are generally very poor therapeutics unless they've been modified in some way. And we use the enzymes to modify the peptides to give them, say, a longer half-life or more selectivity for a specific target, or the ability to start moving towards oral bioavailability. All of these things cannot be done really with the same efficiency if the peptide's linear.
Maybe I'd like to understand a little bit just to clarify, because people, when they think of peptides, I believe they think about anti-aging, they think about muscle building. And so those type of peptides that maybe people are using versus the peptides that you're using or that you're creating are basically those similar peptides, but with the addition of enzymes in order to make them more effective. Am I saying that correctly?
That's a good way to establish a baseline. The neat thing about peptides is they are target agnostic and they are indication agnostic. So it doesn't matter what the target biology is, you can make a peptide that will go after it. So what people don't really appreciate is that you have peptides in your body that are hormones. You have peptides in your body that interact very specifically and selectively for certain receptors in your brain. Insulin is a peptide, and you have all of these other metabolic peptides that help control different aspects of hunger, which is why, the GLP-1s are so popular right now. So what people might not appreciate is that peptides already are incredibly diverse in what they do, what they target, and how they interact with the body. But most of the peptides that your body makes are extremely short-lived. You get a short-lived biological response, your body breaks it down, and then recycles the amino acids to be used somewhere else.
At what phase are you currently in the process of developing these peptide therapeutics? Are they in clinical trials or what is the phase?
So we are currently preclinical. what we were doing first is ensuring that the platform and the screening process that we use, that's what we focused first, is as broad, robust, and as unique as possible. So what we set out first to do is say, take peptides that can hit and multiple oncological targets. Take peptides and find hits for inflammation and immunology targets. Find peptides that could address infectious disease. Find peptides that could address metabolic targets. And what we wanted to do was show that if we do our screens, so we're making, the hundreds of trillions of peptides so far, and this is not in silico, this is in real space in real time. Can we make these massive libraries, run those massive libraries over biology of interest, and then pull out peptides that are a hit? And that's what we first set out to do, which took some, there were a few technical hurdles, engineering hurdles, but we've been able to successfully do that. We can find peptides that we know are hits. We found peptides that activate specific biology. And now what we're doing is starting to take some of these candidates from our screens and start moving them towards the clinic. So that means there's additional steps that we need to do for safety, efficacy, and tox. And then on top of all of that, we are ensuring that we're making something that is going to meet an unmet clinical need.
So currently, are peptides being used as therapies in certain areas like cancer or other ways?
A great cancer example is a molecule called octreotide. Octreotide itself is used to treat a lot of the side effects of cancer. But one of the really neat things that you can do is because octreotide is specific for a somatostatin receptor, and there are 5 different somatostatin receptors. If a type of cancer has somatostatin receptor 2 overexpressed, which is what octreotide targets, you can make a radioligand to octreotide, and that's where you can have the octreotide is, say, the homing molecule, and then you have your emitter attached to it, and that will kill the cancer. So it becomes a very specific way of targeting an oncological space with a highly specific warhead.
So you're creating a database where you have identified which peptides can address certain issues, sicknesses, diseases, and even within that specific disease, maybe which genetic type.
That's one way to think about it. A really good bird's eye view would be, let's take oncology, for example. We know that, say, target X or target Y, say it's HER2 or FGFR, are expressed in very high amounts on cancer cells. What we can do is generate a massive library of peptides that are all enzymatically modified, run that library of peptides on the oncological target, find the peptides that are highly specific for that target, and then move those forward for development. So when you start thinking about things like genetic differences or, say, receptor differences, that is one area that peptides can address. If there's a specific mutation and it starts getting more towards personalized medicine, which is still a far off future, peptides are something that can address that, because you can start making your screens be tailored to the mutant and not whatever the normal form is.
What inspired you to start Sethera?
Well, so I was really drawn to this because I like that what we're doing is a combination of, a lot of scientific curiosity, but then the possibility of real world impact. One of my hangups with academia, even though I love it and I appreciate that it's constantly furthering human knowledge, is that I think that a lot of results and publications in the academic space typically don't have significant and meaningful real world impact. Yeah, it's great to publish papers. It's great to further human knowledge. And I'm glad that that continues. But I really wanted to start moving towards something that I knew can impact human biology and in a very positive way. So what we're doing is we're building new molecules. That's really what it is. Molecules that have never existed before in nature. And that I'm just glad that biology or the enzymes allow us to make things with almost unimaginable complexity. but now we're trying to control that complexity. Enzymes are fantastic because that's where chemistry and biology really meet. They're a molecular machine that performs very complex reactions with a level of precision that's absurdly difficult to reproduce synthetically. So I love that we're able to explore new enzymatic space. We're able to start building molecules that are new, and we have a direct way of translating those new molecules to impacting patients and addressing previously undruggable biology.
What is the future of Sethera Therapeutics? What are you looking at in the short term, and what will be happening, and what are your next goals?
So I think peptide therapeutics themselves will become more sophisticated, and we want to be one of the main tools that is enabling that. Historically, they were always viewed as unstable or difficult to deliver, but that view is changing. We have, and Sethera is developing better tools to stabilize peptides, modify them, screen them, and understand how they behave in biological systems. I think that we'll start to see more of a convergence between high throughput screening, enzymology, synthetic biology, and machine learning. All of these pieces that we're pulling out of screens, we're sitting on terabytes upon terabytes of data that we can then start using to make better AI, better ML. And that's one of the things that I really like about what we're building is we are upstream of all AI and ML. We are generating structures that have never been part of any screening campaign, have never been seen before in nature. So the only way to understand what they are, what they do, is to, look at what we're building. So I think broadly, the next phase of the field will be going after more of these targets that were difficult or undruggable. So for example, if you have, say, a cancer tissue or a tumor that is you can't penetrate with a biologic, and then a small molecule can't actually address it well enough. Peptides can be cell penetrant. They can be extremely target specific. So you can get into this space where small molecules can't do it adequately, biologics can't do it adequately. Peptides are going to be an increasingly popular modality in the future because of what they enable.
Are you trying to produce those and to go clinical, or will it be all preclinical?
We are actively going to start moving our molecules towards the clinic. That is a focus that we have now. The baseline foundation was ensure the screening platform is as robust as it can be. But now that we've developed it and we feel confident in it, we are now moving molecules from our screens towards preclinical studies and IND enabling work.
What you're doing basically is making those existing peptides much more effective and having the information from this library about where they will be particularly effective.
Think of it like this. If you have a noodle
Mhm.
and you want that noodle to fit a very specific shape,
Mhm.
and that specific shape is like, say, the necessary piece to unlock something. So you need a noodle that's like a key. That doesn't work very well, right? Sure, you might be able to twist and contort the noodle into the right shape, but it won't stay in that right shape, right?
Mhm.
That's your peptide. That's just a
Mhm.
normal linear peptide. What Sethera does is we can staple the noodle into the right key shape.
Okay.
So what we do is we make massive amounts of uniquely shaped noodles, staple them so they can fit into the right shape and go into the right keyhole. And then we say we have a lock, and we run every single noodle over the lock and find only the noodles that fit the lock.
The existing peptides that people are using, do they have an aspect like your peptides that you're going to be creating with the enzyme or with the technologies that makes it effective, or no?
Yeah. At the simplest form, like we mentioned, a peptide is a chain of amino acids. It's a small protein. Our bodies are incredibly efficient at recycling proteins. So, one unfortunate reality for many of the people that are injecting peptides is if those peptides are linear, they're being broken down the moment that they're injected, which means that there's going to be little to no effect. And I think that that's probably a good thing in some ways, because peptides themselves can have very potent effects on biology. And so that's sort of one of your body's main ways to protect itself, right? Peptides need to be modified in order to last long in biological media. So the peptides that are in skin cream, that's very different, because in that scenario, you don't have proteases that are going to break them down as quickly compared to, say, if they were injected into your blood or they got into a cell. So that's a separate area where those peptides are typically specifically made to have a certain shape. I mean, you've heard probably about like the collagen hybridizing peptides. Those help build structure to something that already exists. So they end up being relatively stable compared to the ones that are injected. The Sethera platform can address each of these spaces because we are building peptides that are naturally proteolytically resistant. But I think I would maybe caution the audience, or in general, I've already cautioned my own family who've called to talk about these things. If there's a peptide that you're getting from an unusual source, you don't know how it was purified. Peptide that were made by solid phase, which I'm assuming most of these are, have to be cleaved using high concentrations of trifluoroacetic acid. And you don't know if that's been adequately purified away. And I think that there's a lot of concerns with some of these secondhand peptides because realistically, most of them are probably just snake oil. They'll be broken down rapidly by your body, and they don't have the necessary modifications to last a long time.
And so the difference with kind of more the sophisticated peptides that can be used and that Sethera is working with and has developed the technology to do is utilizing enzymes to deliver those peptides more effectively.
It's not that the enzymes are doing the delivery. It's that the enzymes are building on top of the peptides to enable them to last longer, to enable them to survive, say, going through your digestive system, enable them to, once they're in the body, not be immediately digested back down to amino acids. So we're building upon and protecting the peptides as well as making them more specific for certain biology.
And most likely those kind of secondhand or peptide injections that people are using do not have that enzyme in order to protect it currently.
No, they won't have it at all. The number of people that can produce this enzyme is very few and far between.
Yeah, I think that that is so important. There is so much, as you probably know, in the media, in social media about peptides. It's kind of crazy. And I haven't heard a lot from either medical experts or academia that to be able to give more information about it, or that it exists on, be responsive in social media.
I think we can break it down really quickly into several areas. There are the branched chain amino acids that bodybuilders like to take. That will help you build muscle mass. You need peptides to build muscle. They will be digested into individual amino acids, but that can be used as a beneficial way to help build muscle. That's why you need protein. That works. There are the peptides that are already used in pharmacological and therapeutic applications. Those are the things like octreotide, like the somatostatin peptides, like oxytocin. So oxytocin is another peptide that is extremely important in labor and delivery. There are peptides like the GLP-1s. There are peptides like insulin. So there are quite a number of peptides that already have fantastic therapeutic uses. But those peptides have the appropriate narrow windows, and in many cases have had specific alterations of their structure to enable them to last long in the body. The example here is the GLP-1 peptides have a fatty acid appended to a specific residue. And if that fatty acid is not present, the half-life craters to minutes. Now, there are other peptides that are typically being pushed in social media that I mentioned probably account to, in most cases, snake oil, because their half-life is so short. Sure, you can inject them. Sure, they will exist in your body, but they will be rapidly broken down, and it's extremely hard to tell if there's going to be any effect at all, which is why they haven't gone through the FDA.
So what are the next steps for Sethera in terms of being able then to become clinical?
The next steps are and this is what we are doing, is we are taking the peptides that we have pulled from our screens. We are doing initial tests on cell models. We've moved certain peptides beyond that into animal models, and then we're moving beyond that into showing that we can have a repeated use case where the peptides address biology successfully in a safe way. So that's really the next steps. We've done the screens. We have candidates. We've moved certain candidates into cell models. We've seen successful binding, successful activation. We're then taking some of those peptides into animal models and showing that the biology that is anticipated is what we see.
Karsten, it was such a pleasure to talk with you and hear about what you're doing at Sethera Therapeutics.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.